Hi everyone, Dr. D here, and in this video we're going to be finishing off Chapter 6. So let's go ahead and get started. I think where we left off, we were talking about energy coupling. And what you need to understand about energy coupling is that this is the process by which the energy that is released by an exergonic process is used to power or drive an endergonic process. So let me show you just as a review. An exergonic process is like this example at the top right here. This means a negative delta G process, right? And an endergonic process is a positive delta G process. Notice that exergonic processes release energy. Endergonic processes require energy. So why not, if you want to, let's say you're a cell and you want to accomplish something that's endergonic. What does that mean? Let's say the cell wants to make a chemical that it needs, but that process of making that chemical from reactants to products, it's a positive delta G reaction. Well, how could you possibly do that if you're a cell? You require energy to do that. Well, why not use some other process that releases energy, use an exergonic reaction that releases energy, and then use that energy as the energy you need to drive forward an endergonic reaction. So energy coupling is using the energy released from an exergonic reaction or negative delta G reaction to power an endergonic reaction, a positive delta G reaction. So let's look at an example here. And this example involves ATP. Okay, so ATP is a high energy molecule. Remember chemical energy? Chemical energy is a type of potential energy based on what? Based on structure. This is a high chemical energy molecule because it has high structural energy. And the reason it has such high energy, one of the reasons anyway, one of the main reasons that you should know about is these negative charges. Look, ATP has 3 phosphate groups. That's why it's called TP, triphosphate. By the way, side note, ATP is adenosine triphosphate. We know what adenosine is, right? We talked about that before in Chapter 3. Look at this molecule real quick. Does it look familiar at all? Think about it. Does this molecule look familiar to you? It should. Look what you have here. Is this not a 5-carbon sugar? In this case, ribose. And is this not adenine? Remember A's, G's, C's, and T's? This is A, right? Adenine. It's a pyrimidine, or I'm sorry, it is a purine. It has 2 rings. It is a purine. So this is adenine, the purine. This is ribose, the 5-carbon sugar. And then you have 3 phosphate groups. It's called triphosphate. So this is essentially a nucleotide. That's why it's called a nucleotide triphosphate. It's basically the A from RNA, isn't it? Except instead of 1 phosphate group, you have 3. That's why it's called triphosphate. Tri means 3. Now, that was just my side note fun fact for you, and that is that ATP, the energy currency of a cell, the energy molecule of a cell, is the same molecule you use minus 2 of its phosphates to build RNA, just as a fun fact for you to know. So now that we know what ATP looks like, why is it such a high potential energy molecule? Do you remember that example I gave you with my magnets? I don't know if I still have my magnets around here. I don't think I do. But do you remember that example with the magnets? You got the magnets the wrong way around, and one can go flying across the room because they're the wrong way around. Well, there's a very similar situation going on with these phosphate groups. What charge do these phosphate groups have? I'm pointing to them right now. Negative charge. Do you think, based on what we've learned so far, that a negative charge wants to be right next to another negative charge, which is right next to another negative charge? Or is it opposite charges that come together? Opposite charges come together. The same charge does not want to be butt up right next to another same charge. Does that make sense? So what that means is because you have a negative charge right next to another negative charge, there's going to be repulsion, okay? And these phosphate groups are champing at the bit to get apart from one another. And by the way, champing at the bit is the proper way of saying chomping at the bit. You can look it up. Anyway, another fun fact for you. So think about it. These phosphate groups are right next to one another. They do not want to be right next to one another. So what happens if you hydrolyze the terminal phosphate? What does that mean? That means use water, see? Use water to attack the bond between the last two phosphate groups. You see what that does? That releases one of the phosphate groups. Now you have what's called ADP, adenosine diphosphate, because it has two phosphate groups. One phosphate group went flying off. Why do you think that phosphate group goes flying off with so much power? Well, one of the main reasons, like I said, is because negative charges don't want to be next to each other. And when you hydrolyze it, you give this phosphate group its freedom, and it is more than happy to leave, and that actually releases a lot of energy. Okay, so that's why, one of the main reasons why there's so much energy released and why ATP is such a high energy molecule. Okay, so by the way, do you see this phosphate group? You see how it's called PI? If you're ever wondering, why is this phosphate group called PI? What does that mean, P? What does the I mean? I know what P means. That means a phosphate group. What does the I mean? The I means inorganic. Okay, the I means inorganic. And so this PI means inorganic phosphate. What does that mean? You guys know what organic means. We talked about it. Organic means contains carbon, right? If something has carbon attached to it, it's organic. If something doesn't have carbon attached to it, it's inorganic. So look at this. Look up here. When this phosphate group was attached to the rest of the molecule, was it an organic phosphate or an inorganic phosphate? Well, look. You have to ask yourself, is this phosphate group attached to any carbons at all? So if you follow the bonds, look, there's a carbon right there. There's a bunch of carbons. Remember, each one of these vertices is a carbon, unless it's denoted N or something else. So this phosphate group would be called a organic phosphate because it's attached to a molecule that has carbons in it. Whereas this phosphate group down here, once the phosphate group is released and it's all by itself, imagine if you cut this phosphate group off. Imagine if you cut that off. That's what this is, inorganic phosphate. Now it's called inorganic phosphate because there's no carbons at all in any way attached to this phosphate group. Just as a side note, so you understand what that means. Okay, so why do I show you this? Because a lot of times, most of the time, that your cells need to do something, and that something requires energy. It's an endergonic process. It's a positive delta G process. How do the cells pull it off? Well, the cells will pull it off by grabbing an ATP molecule first, hydrolyzing the terminal phosphate, that means this last phosphate group, and that releases energy. So it's an exergonic reaction that releases a bunch of energy. And guess what the cell does with that energy? Guess what the cell does with that energy? It will use that energy to do work, okay? To do the thing it wants to do. So here's an example. This is one example they picked out of a million different examples that they could have picked. But look at this example. You don't have to get bogged down in the details of this example, but what I want you to understand is the concept behind this example, okay? So look, let's say the cell wants to make this important amino acid called glutamine. You see? Glutamine. How do you make glutamine? How does a cell make glutamine? Well, it takes the reactants, glutamic acid, and then it also takes ammonia, and it has to join the ammonia to the glutamic acid in order to make glutamine. You see that? Now, is that a free process? Is that an exergonic process that's going to happen spontaneously? Well, let's look at the delta G of the reaction. The delta G of this reaction here is, oh, it looks like it's a positive number, positive 3.4 kcal per mole. You don't have to worry about the kcal per mole part. That means those are units of energy. So, but notice how it's a positive number. It's positive 3.4. You know what that means? Is that spontaneous or non-spontaneous? That's non-spontaneous, which means it requires energy. If you have to put some energy into this to make the product glutamine. Now, again, if the cell wants to make glutamine, is it just going to throw its hands up in the air and say, well, I guess I can't make glutamine now because that's not a spontaneous process. It's going to require energy, so I guess it's impossible. No. You know what the cell does? Let's skip to the bottom here. You know what the cell does? It says, hey, I want to combine glutamic acid and ammonia, and I want to make glutamine, but that costs energy. What do I have around here that releases energy? Oh, I got this handy-dandy molecule called ATP. And guess what? When I hydrolyze the ATP, that actually releases a bunch of energy. Look how much energy is released when you hydrolyze the terminal phosphate and release it as inorganic phosphate. Look how much energy is released. That's right. It's like minus 7.3 kcal per mole. So if you did the math over here, you see at the bottom left the math, you needed 3.4 kcal per mole to do what? To make glutamine. You provided that energy by hydrolyzing ATP. That released 7.3 kcal per mole. So if you do the math, you've released more than enough energy to drive the reaction forward. You see, you've released so much energy that you have extra energy released, and that extra energy kind of dissipates as heat. Okay, so again, this is an example of what concept? This is an example of energy coupling. You look what you did. You used the energy released from hydrolyzing ATP, so the energy released from this exergonic reaction, you use that energy to do what? To drive forward a endergonic process. In this case, the endergonic process was making glutamine, you see, and it required energy, of course, because it's an endergonic positive delta G reaction. So I hope you understand what this means. Anytime the cell wants to do something like transport stuff against the concentration gradient or move stuff across the cell, it's going to grab an ATP molecule and hydrolyze it into ADP and inorganic phosphate, and that's what's going to provide the energy to do that thing. Okay, and this is usually how the cell powers these types of things. Okay, whether they be like I said here earlier, whether they be chemical work, transport work, or mechanical work, ATP is the universal molecule that allows those things to happen. It's not 100% of the time, but cells usually use ATP to make those types of things happen. Okay, so this example here is trying to do chemical work, doing energy coupling for making a chemical called glutamine. Here in this example, you're using energy coupling to transport stuff. And then here in this example here, you're using ATP through energy coupling to move stuff. You see what's happening here? There's a vesicle full of cargo. There's a molecule. In this case, it's like an inverted Y. And that's a motor molecule. It's a protein that walks, but it walks. It has this mechanical walking motion with the power of ATP, so it can transport that vesicle where it wants to go. It's kind of neat. We're going to learn all about this stuff later on. Okay, so before we go on to the concept of enzymes and how enzymes work, let me take a quick break and come back. Hey everyone, welcome back from break time. So where we left off, I was about to tell you about enzymes. Enzymes are usually proteins, though sometimes they could have RNA components. But usually when you're dealing with an enzyme, it's a protein, and it's a special kind of protein. Enzymes are proteins that speed up reactions, speed up chemical reactions, and the term for those types of agents that speed up chemical reactions are catalysts. Okay, catalysts speed up reactions, and so enzymes are catalysts. They speed up a reaction, and the way they do that is by lowering the activation energy. I'm going to explain what that means in a little bit. Another thing you need to understand is that catalysts function by speeding up a reaction, but they don't get consumed by the reaction. What does that mean? The reaction can happen over and over and over again. Okay, so an enzyme is in essence a catalytic protein. Okay, usually a protein, like I said, and once in a while RNA as well. If it's an RNA enzyme, it's called a ribozyme. Now, let's go on to an example. Enzymes usually end in the suffix ase, A-S-E, ase. So for example, sucrase is the enzyme that breaks down the substrate. The substrate is the thing the enzyme acts on and speeds up the reaction for. And the substrate for sucrase is sucrose, the disaccharide. And you guys remember what sucrose is? It's a disaccharide of glucose plus fructose held together with a glycosidic bond. Now, sucrose breaks down into glucose and fructose, and this is a spontaneous reaction where water attacks the glycosidic bond, separating the two from one another with a hydrolysis reaction. However, if you were to add sucrase, the enzyme, then this process is sped up manyfold. Okay, if you remember from Chapter 6 lab, the important lab where you're going to do a lab report with the results, Chapter 6 lab dealt with the enzyme peroxidase. See how it ends in ase, A-S-E? A lot of enzymes end in ase, though not all do. Peroxidase is the enzyme. Do you remember what the main substrate was for peroxidase? Hydrogen peroxide. Usually the enzyme's name gives you an indication as to what is being broken down by that enzyme. So if it's sucrase, it's breaking down sucrose. If it's peroxidase, it's going to break down peroxide or hydrogen peroxide, to be more specific, okay, into water. So in this case, sucrase is breaking down sucrose into glucose and fructose with a hydrolysis reaction. And do you remember what I said earlier? I said catalysts function, or enzymes function, by lowering the activation energy. This is how their reactions are sped up, by lowering activation energy. So what is activation energy? Activation energy is the initial energy needed to start a chemical reaction. Okay? A lot of times what you need to understand is often activation energy is supplied in the form of thermal energy. So heat. Heat can serve as activation energy. And usually heating something will speed up the reaction. You guys are familiar with that. Sometimes you can't seem to dissolve this in water, or you can't seem to get this reaction to go. You heat it up a little bit, or you provide a spark, and that spark or that heat is the activation energy needed to start a reaction. Even if it's a spontaneous reaction, it will make it happen faster. Okay, so let me show you what I'm talking about here. Let's say we have an exergonic reaction where the reactants are at a high free energy. Remember, free energy means G, capital G, or Gibbs energy. The reactants have a high free energy and the products have a relatively low free energy. So what kind of reaction is this? Where you're going from high G value to low G value products? That would be a negative change in G, because you went from a high G to a low G. This is the progress of the reaction on the x-axis. And so if you're going from high G to low G, like in this example, it's a negative change in G, a negative delta G reaction. Okay, so this is a spontaneous reaction, right? Because this is an exergonic reaction. Exergonic reactions are, by definition, the reactants have a higher free energy, the products have a lower free energy, and there's going to be energy released, by the way. Remember, there's going to be energy released by this process because it is an exergonic or negative delta G reaction. So what is this speed bump then? What is this black line, and why does it go up into this hill? You know, that what is this speed bump? I call it a speed bump, an energetic speed bump. That's the activation energy. Remember what activation energy means. The initial energy needed to start a chemical reaction. So do you guys remember when I said that just because a reaction is spontaneous, it doesn't necessarily mean it's fast? That's what I meant. If a reaction is spontaneous, all that means is the reactants are higher free energy than the products. But what makes a spontaneous reaction fast or slow? It's this. It's the activation energy, the initial energy needed to start that reaction. And if this is a big energy requirement, if this is a big speed bump, a big EA, activation energy, then it's going to be a very slow process. And if it's a small bump, if it's a small EA, then it's a very quick process. That's what determines whether a reaction is fast or slow. Bigger EA, slower process. Lower EA, faster process. Okay, so how could you provide EA? A lot of times you could provide the activation energy by heating up the reaction. But what if you don't want to do that? Well, that's where enzymes come in. Look, if the black line is the activation energy without an enzyme present, then the red line would represent the activation energy with enzyme present. Do you see that? So when you add enzyme, it lowers the activation energy and speeds up the reaction. Okay. So let me just show you real quick what this process looks like. You have an enzyme, and an enzyme is usually a protein. And remember what proteins look like? They have this globular structure. Why? Because remember, proteins are just one long chain of amino acids which fold into secondary structures like alpha helices and beta sheets, and then those fold into a three-dimensional structure called the tertiary structure. So this blue-purple structure here, this would represent the tertiary structure of a protein. And do you remember when I said a protein must be folded correctly to function? You see why that's important? Because if you have the correct folding, you're going to have the correct active site. The active site would be the correct three-dimensional shape that accommodates the substrate. The substrate fits into this three-dimensional shape called the active site. And now you have what's called the enzyme substrate complex. And in this case, the enzyme is doing an induced fit, which means it's locking that substrate in place. It's going to then act on that substrate. So for example, if this enzyme was peroxidase, the substrate would be peroxide or hydrogen peroxide. If this purple thing was sucrase, this substrate would be sucrose. The enzyme and the substrate come together, they lock together, and then the enzyme can catalyze some kind of reaction on the substrate. Okay, and remember what the enzyme's doing? The enzyme is lowering the activation energy requirement for the reaction to take place. Okay, so again, you have a substrate, it binds to the enzyme. So for example, if this was sucrose, it's the substrate for the enzyme sucrase. You then have an enzyme substrate complex. The enzyme then lowers the activation energy, allowing the enzyme to catalyze a reaction on the substrate, and then the substrate releases the products, the catalyzed products. Now, you might be wondering, okay, I understand activation energy is the initial energy required to start a reaction, and enzymes function by lowering the activation energy, which therefore speeds up a reaction. But what does that all mean, Dr. D? What does that mean, really? That sounds good, but what does it actually mean to lower the activation energy? Can you show me an example of an enzyme doing that, or what that would mean? Yes, let me show you. These four right here, these are examples of exactly how enzymes might lower activation energy. So in case, like I said, in case you're wondering, that sounds very hand-wavy. That sounds very vague and ambiguous. So what do you mean lower activation energy? It is not very solid. It's, can you give me an example? Well, here are some examples. Enzymes can orient substrates correctly. That could lower the activation energy. That could help break down the substrate into product. It could strain the substrate's bonds. The enzyme could provide a favorable microenvironment. For example, the active site could be an acidic active site. It could be a basic active site. It could covalently bond with the substrate. All of these might lower the activation energy. So let me show you an example. Look at this. Let's say this is the enzyme in blue at the bottom. This is the enzyme, and look, it's the enzyme sucrase. Remember, sucrase. What's the substrate for sucrase? The thing that it binds to and then catalyzes a reaction? This up here. This is sucrose. Sucrose, a disaccharide. Here's the saccharide glucose. Here's the saccharide fructose, right? So, sucrase binds to sucrose. There's the enzyme, the enzyme substrate complex right here. And look closely here. Look closely at what the enzyme is doing to the substrate. Look at this. Look here. Do you see what I'm highlighting right here? Look at that. This is the glycosidic bond between glucose and fructose. Okay, this is the glycosidic bond. Okay, and by the way, I don't know why, but they drew fructose with as a 5-carbon ring when it's actually a 6-carbon ring. It's strange. Anyway, but let's focus on the task at hand here. Look what the enzyme's doing to that glycosidic bond, the bond between the glucose and the fructose. Look at this. Notice how the bond's perfectly straight, the glycosidic bond's straight. What did sucrase do to that bond? Do you see how it's now bent? Do you see how it's now bent? The glycosidic bond between glucose and fructose was straight, but upon binding to sucrase, it bends. So, do you see what sucrase did? Sucrase got in there, grabbed the substrate, and gave it a bend. And that bend, look what it does. It strains the bond, and it exposes it better to water, so that the water could hydrolyze that glycosidic bond and break it apart into glucose and fructose. What example was that? Do you see that example here on this list of how enzymes lower the activation energy in a concrete fashion? Yeah, if you said it's an example of straining the substrate bonds, you'd be right. So look, here sucrase is straining that substrate bond, that glycosidic bond, allowing water to more easily hydrolyze and attack that bond to separate the glucose from the fructose and produce the products. That's how sucrase acts as an enzyme. It's lowering the activation energy by bending the bond. Now, how would you speed this reaction up? You know, where sucrose becomes glucose and fructose. How would you speed this up without enzyme, without sucrase around? Let's say you didn't have sucrase around. Well, remember what you could do? You could just turn up the heat. Right? Turn up the heat. You know what that would cause? That would cause this molecule to kind of bounce around. Water would be more likely to hit just the right spot on the sucrose, and that would also help speed up this reaction. So remember what I said earlier, sometimes you could provide the activation energy by heating up the reaction or providing a spark or flame or something, and sometimes you could add an enzyme, and that lowers the activation energy to speed up the reaction. And do you remember what I said as well before? Enzymes are usually proteins that look like this, right? Enzymes are usually proteins. Remember I've been making a big point that proteins need to be folded just right to function. And do you remember in lab 6 in the lab, we looked at how environmental factors can affect the folding of a protein and an enzyme, peroxidase, things like pH, things like temperature, and things like ionic conditions. These can affect the folding of a protein or folding of an enzyme. Let's say it's a enzyme from your cells. If you were to then boil that enzyme, what do you think would happen to the nice three-dimensional structure of that enzyme? Well, it would denature. Do you remember proteins can denature? So extreme shifts in temperature towards boiling, extreme shifts in pH, all of these things can cause proteins to denature. And once denatured, those proteins do not renature on their own. They do not renature. So you actually destroy an enzyme's function and a protein's function for that matter by changing the temperature too high or changing the pH too acidic, too basic. Now, what you do need to understand is real quick, this is actually an interesting point. Not all enzymes work at the same temperature at the same pH. I just want you to realize that. For example, look at your own body. You guys know how you have enzymes in your stomach? You have a bunch of enzymes in your stomach that help digest your food. Do you think those enzymes fold best at pH 7, the normal pH of like a neutral pH, right? Do you think those enzymes fold at pH 7, or do you think those enzymes would fold more best at pH 1, closer to the pH of your stomach? Well, the answer is the enzymes that are inside of your stomach, they actually fold best and work best at acidic pH, at pH 1. However, what about the enzymes in your skin or in your brain or in your heart? Do you think those are best folded at pH 1? No, those are going to be best folded at a more neutral pH range around your physiological pH. That means the pH of your body and your fluids, right? So just be aware of that. Not all proteins fold best at the same temperature, but all proteins have an optimal temperature for folding and an optimal pH for folding. So it's good to understand. All right, now here's another thing about enzymes that's really interesting, and it's called cofactors. A lot of enzymes require little helpers to work correctly. Okay, so without these little non-protein enzyme helpers, the enzyme may not either fold correctly, so it can't function correctly, or sometimes these cofactors are part of the active site of the enzyme, or they help catalyze the reaction of the enzyme. So what you need to understand is that your enzymes may or may not require cofactors, and these cofactors are important enzyme helpers, and they're non-protein enzyme helpers. So what do I mean by a non-protein enzyme helper? Let me give you an example. You guys know of the protein hemoglobin. We talked about it earlier when we discussed hemoglobin and sickle cell anemia. Did you know that hemoglobin requires the cofactor iron? Have you ever heard that you need iron in your blood? Everyone's heard that, but what does that mean? Well, that means that literally your red blood cells have iron in it. That's why blood has a faint smell of iron, okay? Iron is a cofactor for hemoglobin. Without iron, hemoglobin doesn't function correctly to carry oxygen, okay? So iron is a cofactor for hemoglobin protein. Does that make sense? That doesn't mean all proteins and all enzymes require cofactors, but some enzymes like hemoglobin do. Have you ever heard of iodine as a cofactor? Iodine? Well, without iodine, you're going to have thyroid issues. You're going to have goiters and things like that. You're going to have all kinds of thyroid issues if you don't have enough iodine in your diet. So you see iodine, iron, these are minerals, aren't they? You know, the elements, the minerals, iron, iodine. Other cofactors might be calcium, cobalt, zinc, magnesium, manganese, even silver and gold, those types of things. All could be cofactors. So cofactors could be minerals. Minerals are like different elements, essentially, right? Those cofactors would be inorganic. Okay. Other cofactors can be organic, and here's what you need to understand: the organic cofactors are called coenzymes. Organic cofactors are called coenzymes. Inorganic cofactors are not called coenzymes; they're just called cofactors. And what are coenzymes? Coenzymes include these guys, the vitamins. Okay. Vitamin A, folic acid, vitamin B1, vitamin C. You see, all of these are examples of vitamins, which are coenzymes. So what does that mean? It's It's a enzyme helper, a non-protein, none of these are proteins, a non-protein enzyme helper that has carbon in it. See, all of these have carbon in them because remember I taught you how to, I'm sorry, I taught you how to decipher these codes here, that each vertice, each point is a carbon, right? So there's carbon in all of these molecules here. So have you ever heard of, getting your vitamins and minerals, or this breakfast is a good source of vitamins and minerals? And, you know, in general, since you were a child, you focused on getting the right vitamins and minerals in your diet. Well, you ever wondered why those things are important? Now you know. Vitamins and minerals are important cofactors and coenzymes for different proteins and enzymes inside of your body. And again, not all proteins require cofactors or coenzymes, but those that do really do. And that's why you can't have a diet free of iron. You can't have a diet free of iodine. You can't have a diet free of even cobalt, because that cobalt might be required by a particular protein in your body, and I'm sure of it, that it is. Okay, so that's an interesting thing to understand, right? That's an interesting thing, and now you know why you need vitamins and minerals in your diet, because they allow certain proteins and enzymes in your body to function. Fun fact: do you know where we get most of our iodine in our diet? We get it from iodized salt, right? They found out a long time ago that it's actually quite difficult to get enough iodine in a diet, right? So they said, well, let's add the iodine, let's supplement the iodine in people's food, and they figured, well, what does everyone eat? And they said, well, salt is pretty plentiful in people's diets. Well, then, okay, let's add the iodine to the salt. And that's what they did. They added iodine to salt. That's why when you go to the store, you see iodized salt, because you need iodine in your diet. And unless you have a diet rich in fish and some other things that are really not all that common, then you're not getting enough iodine. You're going to have those thyroid issues. And so that's why there's so much iodized salt. Fun facts. All right, moving on. So if a cofactor is an enzyme helper, inhibitor is the opposite. An inhibitor, the term inhibit means to stop, right? Or cease. An inhibitor is a molecule that binds to an enzyme and stops it from working, okay? A substance that binds to an enzyme and decreases or stops, attenuates its activity. Okay, now these inhibitors, these molecules can be called competitive inhibitors, or they can be called non-competitive inhibitors. Let me show you. Let me show you what these inhibitors look like. So if this is the enzyme in blue, and this is the substrate in, what is this, like a burnt orange? Well, the inhibitor would be in green. You see what the inhibitor is doing? In the case of competitive inhibitors, the inhibitor binds to the actual active site of the enzyme, and it does so by mimicking the substrate in some way. Think about this. If you have an inhibitor, this imposter molecule, this green thing, and it's stuck to the active site of the enzyme, do you think the substrate can bind to the active site and be broken down or whatever it needs to happen to it? No. That's why it's called competitive inhibition. The inhibitor is competing for the active site of the enzyme, and it's out-competing the substrate. This poor substrate can't get in there and be broken down properly. You see that? So if this was sucrase, for example, this blue one was sucrase, and this orange substrate was sucrose, the sucrose wouldn't be able to get into its active site and be broken down into glucose and fructose. You see how that works? So inhibitors, like I said, competitive inhibitors usually mimic the substrate. That's why they're able to lock into the active site of the substrate. The active site of the enzyme, because they're mimicking the substrate, and so they're occluding, they're blocking entrance to the actual substrate. But what about this example over here? This is kind of weird. Look, in this case, look what the inhibitor's doing. The inhibitor's not binding to the active site. In this case, in non-competitive inhibition, the inhibitor is binding to a site other than the active site. You see, it's not binding to the active site, it's binding somewhere else. However, that still has an effect on the active site usually, and that still has an effect on substrate binding or breakdown of the substrate. So this is called allosteric regulation. This is the allosteric site, allosteric regulation. Let me explain something. Do you remember what these enzymes are, this blue stuff is? Do you remember what the enzymes are? It's basically a long chain of amino acids, right? And that chain of amino acids has to fold perfectly into this three-dimensional structure. So imagine if my arms are a protein right now. You see, my arms are a protein. Well, is it my hand connected to my elbow, right? You see that? Because the protein's one long chain of amino acids, right? So what's one way of disrupting my active site? Let's say my hand was my active site. Well, one way of disrupting it would be stuff something in the active site, right? That would be called competitive inhibition. But what if I stuff something on my elbow? What if I jam something on my elbow? What's my elbow connected to? It's also connected to my hand because everything's interconnected. So if I were to jam an inhibitor onto my elbow, I could theoretically mess up my hand as well. And that's kind of how allosteric sites work. It's a site where inhibitor can bind but still have an effect at a distant site. So let me define this again. Allosteric regulation occurs when a regulatory molecule, such as an inhibitor, binds to a protein at one site and affects protein's function at another site. Why would that happen? Well, because, again, proteins or enzymes are long chains of amino acids that have to fold just right. If it's all folded just right, it's all interconnected, you don't have to, for example, you don't have to tinker with the active site to tinker with the active site, because if you mess with my elbow, you might mess with my hand, right? Okay, and this. Oh, by the way, what types of things are inhibitors? Real quick, let's look at this. Here, examples of inhibitors are toxins, poisons. You know, those things from venomous snake bites and things. Pesticides, antibiotics. These are all examples of inhibitors. But your cells can also make inhibitors on purpose. Like, not all inhibitors are poisons or toxins. Sometimes your own cells will make an inhibitor for a good reason, not to harm you, but for a good reason. And an example of that is called feedback inhibition. This is the final concept of Chapter 6. Let's talk about it. So feedback inhibition. What is this feedback inhibition? Before I tell you exactly what feedback inhibition is, let me tell you a scenario where feedback inhibition might be useful. Okay, just let me give you a scenario where feedback inhibition would be useful before I even tell you what feedback inhibition is. Okay, real quick, just bear with me. Look at this picture here. In this picture, you're starting with a blue substrate, right? And they picked the substrate threonine. They could have picked 1,000 different examples. Okay, don't get bogged down with threonine. Like, I think I have to memorize threonine. They picked threonine because it's an example of a feedback inhibition mechanism. But here, look. Let's say you have a substrate threonine, this blue stuff. And this blue stuff's handy. It's important, right? Threonine is an amino acid that your body needs. And then imagine if a bunch of enzymes, you see here enzyme 1, were to convert threonine into some intermediate A, and then another enzyme could pick up where that left off and convert that intermediate A into an intermediate B, and then a number of enzymes, you see a number of enzymes are converting that initial substrate threonine into this final end product called isoleucine. Okay, did you see what happened there? We converted the blue stuff into the red stuff. Okay, we converted threonine into isoleucine. Isoleucine is another amino acid. So, okay, think about it. If threonine is an amino acid and isoleucine is an amino acid, don't you need both of those amino acids? Remember, there's 20 different amino acids. And look what this metabolic process is doing. It's converting this amino acid, threonine, into this other amino acid, isoleucine. Now here's the scenario I want you to understand. Do you think your cells want to convert all of the amino acid threonine into isoleucine? Do you think it would make sense to convert all of this blue stuff into red stuff? In this case, it may not be the best thing to do. You know why? Because threonine is an important amino acid, but isoleucine is also an important amino acid. Do you see the problem there? If you were to convert all of the blue stuff into red stuff, the red stuff's great, but you'd run out of blue stuff. You wouldn't want to do that. Do you see what's happened here? Are you following me? What I'm trying to say is, if a cell wants to convert an initial substrate into a product, but not all of it, but have some initial substrate left over, because the initial substrate, you don't want to convert all of this to this, you just want to convert half or something. That's a prime ideal scenario to employ feedback inhibition. That's why cells would need something like a feedback inhibition. So what is feedback inhibition? Let me explain to you what that means. You know what that means? Look, when you convert an initial substrate through a number of enzymatic steps called the metabolic pathway to an end product, that end product can actually feed back and serve as an inhibitor for the very enzyme that started its production. You see that? So in this case, again, what's happening? Your cells want to convert the blue stuff into red stuff, but not all of the blue stuff. So what happens? Some blue stuff gets converted to red stuff. The red stuff accumulates until it reaches some threshold level. At that point, the red stuff itself, it comes back as an inhibitor, which can block enzyme 1. And if the product itself can feed back and block its own production, that's feedback inhibition. And when would you want to employ feedback inhibition? When would cells want to do feedback inhibition? They would want to do feedback inhibition anytime the initial substrate is important or of value and only a limited amount of the end product is required. Now, if a cell didn't care how much of this threonine became isoleucine because only the isoleucine was important, then you wouldn't need a thing like feedback inhibition. You wouldn't have to use feedback inhibition. So hopefully that makes sense. Again, don't get bogged down in threonine becoming isoleucine. That's one example out of thousands that they could have picked, but it's a good example. That's why they picked it, because you need both amino acids. You don't want to convert all of one into another. That's just as bad as not having the one that you made, right? So anyway, that's feedback inhibition, and that leads me to the end of this chapter. Thanks for watching all 3 parts. Hopefully it made sense of a very difficult concept of metabolism, thermodynamics, enzyme dynamics, enzyme kinetics, and all this. And hopefully it made some sense. We're moving on to chapter 4 next, which is actually a lot more for you visual people out there. And so hopefully we can pick up there later this week. All right, thanks for watching. Let me know if you have any questions, and we'll catch you next time.